My whole career, I have been distressed over the way that people compartmentalize information as if the world were objectively split up into little academic departments. And of course, neither the body nor our body of knowledge work in that fashion. And so what I think is so visionary on Jen's part is that we're hearing about the personal, the political, the dietary, the clinical, and now we'll be hearing about the mechanistic aspects, all of ketosis and the ketogenic diet and how it might influence the brain. So like many have suggested, the distinction between neurological and psychiatric disorders is essentially entirely historical, and I think unhelpful from a scientific perspective and from a clinical perspective. And so in my own career, I have very much followed the questions and not paid too much attention as to what department this might bring me to. Am I using tools from one field versus another? In the end, we use the tools that are appropriate for addressing the questions that matter. And so what I will be talking about today is how I believe those questions ought to be framed, which again is much more integrative than is typical within the field that I work in, and suggests some potential tools that could be used to assist us in thinking more holistically about these disorders. So within the field that I've been working in neuroscience, so I started off my career originally studying mathematical logic, then moved to a department of theoretical physics. And what I really loved about the work I was doing was the whole idea of building a field up from first principles. So certainly in mathematical logic, you start from premises. From those premises, you then derive, let's say, intermediary lemmas. From those lemmas, you then eventually derive your conclusions. And so you can very transparently see the entire structure of the field. In theoretical physics, likewise, it's not quite as transparent in terms of how you derive different elements of the field. But at the same time, there is an assumption that if you could identify those first principles, the rules that govern the behavior of physical laws, then everything else follows. And it seems to me that that approach is largely missing from our understanding of medicine, where people tend to work on one little element or another little element, and the question is, what are the fundamental rules? And so that is my own kind of approach. So in thinking about psychiatric and neurological disorders, my first stop was really thinking about, again, starting from first principles, not assuming too much, but starting from what I could measure. And in humans, if you want to look at the brain, you need to use neuroimaging. So we have many wonderful tools in neuroimaging. Some of them talk about the structure, some of them talk about the function. Increasingly, we have been thinking about not just activation of particular areas in functional MRI, but actually connectivity. And all of these approaches are informed by a particular way of thinking, which historically came out of neurology, which is that the brain is divided up into little regions, and these little subregions are associated with particular functions. And this came out of a history of dealing with people who had brain tumors, so it's a kind of a lesion model. And the question is, can we then understand the brain as a function of how those different regions play a role in performing different tasks that the brain needs to do? So unfortunately, that way of thinking really doesn't work when it comes to perhaps most disorders, but certainly psychiatric disorders. In psychiatric disorders, one thing that is very clear is that we are talking about circuits. Certainly, we know we're talking about circuits because if you look at a structural MRI, you do not see lesions in the brain of a psychiatric patient. When medications do work, those are medications that target neurotransmitters. And so the other thing that you notice, of course, is that when you look at the dynamics, the trajectories of clinical psychiatric disorders, you notice that often there's a precipitating stressor. Something happens. There's a perturbation that for most of us would be mildly destabilizing, but for someone who has a propensity for psychiatric disorder is more than mildly destabilizing. It often induces a first break. And even when there is a period then of remission, often another stressor may be enough to induce a relapse. So even, we're used to thinking about bipolar disorder as being periodic, but in fact, most psychiatric disorders do have some periodic element. And so we need to think about them dynamically and what's responsible for those dynamics. So the way I think about it is that in really every other physiological disorder, when you think about what makes for a healthy homeostatic regulatory system, you have excitatory and inhibitory components of control feedback loops that maintain homeostasis in the face of chaotic inputs. So certainly when we think about how much glucose you should have in your body, our body needs to be able to take in glucose, but then regulate glucose, because too much is a problem, too little is a problem. And so you have all of these feedback loops that actually maintain your energy levels within the ranges that your body requires. And of course, it's not just glucose, it's also ketones, it's also sodium, it's also potassium and oxygen and every other substance in the body. So if you want to think about disorders, you need to think about dysregulation, and dysregulation is a problem of feedback. You'll also notice that when you look at a healthy control and a type 1 diabetic, so this is an invasive glucometer, over a week. And what you'll notice is that it's really not the levels that matter, it's the dynamics, right? And so if you were to measure a diabetic and a non-diabetic person, they might have exactly the same levels of glucose, or the diabetic might have higher levels of glucose than the healthy control, or lower levels of glucose than the healthy control. And therefore, that suggests that really what we need to do, and we're thinking about dysregulatory disorders, is not think about levels, but rather the dynamics of the system as a whole. And of course, there are many other feedback loops, and I will only very briefly touch upon them, but thinking about the regulatory system in the brain is a little bit different than in the periphery. If anything, the regulatory controls are much tighter in the brain because, unlike the periphery, which has access to the liver, which is a large organ, the glucose and glycogen regulation is happening in the glia in the brain. So, ketosis might take 24 to 36 hours to achieve in the periphery, but you go into ketosis, your brain goes into ketosis every single night because you'll deplete your glial glycogen stores within 6 to 8 hours. Okay, we're not going to be able to get away with simply saying, okay, what does the brain look like? So what is the alternative? So I would say if we think about physical models, we have some billiard balls and we have some set of initial conditions, and now we do something to the system and it evolves over time. And so ideally we'd want to have something that looks like this, but ultimately, of course, we're really thinking about homeostatic regulation, which is a way of thinking that comes from control systems engineering. So in short, basically, we'll need to be thinking about dynamics, not levels. Ultimately, for dysregulatory diseases, you want to treat them pre-symptomatically, because by the time you're symptomatic, the system has degenerated to such a degree it will be much harder to correct. You need to consider that dysregulation is not one thing, but potentially very many, and different disorders might emerge from the many different ways in which feedback can get disrupted, both feed-forward and feedback. These are complex disorders. You have not one set of nested control loops, but many that are all interacting with each other. So this is kind of not a trivial one thing goes up, one thing goes down type of situation. And ultimately, we want to be able to measure these control loops in a non-invasive manner, simply because the moment we take them apart, we break them. And so when you break them, you can't study their regulation anymore. So at this point, with that introduction, I'm going to move much faster. So what we're doing is basically developing multi-scale models. This one is based on principles of statistical physics. We start at the very bottom layer, thinking about what's happening at the neuronal, glial, mitochondrial scale. That goes then through a neurovascular coupling function. There's sort of an optimization function that thinks about how networks reorganize, all the way up to fMRI. So we need to think this way because clinically, we're always going to be looking at what the brain is doing, and to do that, we need to be able to use neuroimaging tools like functional MRI. But if we want to intervene and ask what is the root cause of what we're seeing at the top level, we need to have access to the bottom level as well. So here is that model, which you just saw, and you can see that you have the excitatory and inhibitory neurons firing. So it's a dynamic process, and you'll notice that their interactions are changing, right? So we're going to be thinking about insulin resistance and the brain. Not that this is the only mechanism, it's one of many pathways, but it's the one I'll be talking about briefly. And the motivation initially was thinking about brain aging in the context of hypometabolism and whether insulin resistance might be a mechanism that is causing that hypometabolism in neurons. So motivation for this, very briefly, can be found from looking at large-scale data sets. So here we analyzed 45,000 brains through UK Biobank, looked at cognitive function, brain atrophy, brain activity, and so forth. I will simply summarize by saying that brain aging certainly does accelerate in individuals with type 2 diabetes. And when I say accelerate, it doesn't look like it's a different process. It actually seems to be affecting the same systems in the same order, but just faster. So we're going to talk very briefly about network stability. So what I mean by that is if we are looking at the communication between different parts of the brain, we can call that a network. And if we ask how it changes over time, then this will be its stability or instability. So the tau here is the stability over what length of time. And so what we found initially is that if we look at brain network stability over the lifespan, first of all, the brain destabilizes. Interestingly enough, it destabilizes faster than you'd think, starting in the late 40s, around 47. And this is the base of the sigmoid. And so we found pretty consistently that there was this sigmoid-like behavior. So we initially found this in two groups, one of which was 636 brains, and the other one was 292 brains. And then, because I'm a skeptical sort of person, we then replicated with another data set of 725 brains, see the same sigmoid. And because I'm a skeptical person, we wanted to figure out, is this actually hemodynamic? Because fMRI is measuring the hemodynamic response, or is this neuronal? But this replicated also in MEG, which is neuronal. And then if you look at yet another data set, so now we're talking about replicating this in 6 different data sets. This one is separated by APOE4 positive and negative, so this is the greatest risk factor for Alzheimer's disease. And what you will find is the same sigmoid, which occurs in people who are APOE4 negative, so this is something that seems to occur over everyone, is greatly exaggerated in people who have the allele that puts them at risk for Alzheimer's disease. So in thinking through then, how could we intervene? So if this is something that's happening in the late 40s, then that suggests that maybe there's something we might be able to do about it. Obviously, at this point, people are still pre-symptomatic. And so if, in fact, what's happening is there is a problem with GLUT4 transport, perhaps ketones might provide a backdoor for refeeding the brain. So we have then a mechanism in principle of both tightening and releasing energy constraints. Because we were working with beta-hydroxybutyrate, and in fact, we knew the pharmacokinetics in the periphery, but we were the first group to use it in the brain. So we didn't know what the pharmacokinetics was in the brain, or even if it passed the blood-brain barrier. So we started by doing a study in MRS, and we looked here at the ketone ester and glucose over time. This is in the brain. And you can see that it hits its peak after about 30 minutes. And you will also know, by the way, that glucose goes up and then it goes down. And again, we're measuring what the brain is doing. Ketones go up and they're quite stable. So we started by putting people on 1 week of a ketogenic diet. So basically they were tested 3 times on a standard diet after an overnight fast, and then after 1 week of a ketogenic diet. And what we found is actually networks were stabilized. So in other words, I just showed you that networks become destabilized as you grow older. And so what has happened now is in these young, healthy individuals who don't necessarily have anything wrong with them, after 1 week on a ketogenic diet, their brains have stabilized. They are behaving like younger individuals. We then replicated this with beta-hydroxybutyrate. When you put people on a diet, lots of things change, caloric value, the macronutrients, and so forth. So in this way, we were able to calorie match and weight dose the beta-hydroxybutyrate to the glucose. And we also see that there was stabilization of networks. We then looked at what happens when you combine glucose and ketones. So you have someone on a standard diet and you give them beta-hydroxybutyrate. In contrast to the conventional wisdom, which is that glucose is the brain's preferred substrate, in fact, what we found is that given the choice between the two, the ketones won, and we're still stabilizing the network even in the face of a normal diet. So what we want to do then is think about how is the brain, what does it mean to stabilize networks? And in this way, we can take tools from statistical physics that look at what it means to optimize under resource constraints. What we found is that basically there is a phase transition that happens in the late 40s. When people are young, there are basically two types of networks that are activated. The one that is segregated is very local. These are the sort of local processing units. But there's also communication between those sub-networks, and that represents the integrated network. So in a normal, healthy young brain, what's happening is that the brain is constantly switching between the integrated and the segregated, but that what happens past this critical point is that the brain gets sort of stuck in a segregated regime where it is not able to communicate as effectively between different brain regions. So we'd like to understand why, so we started looking mechanistically at the bottom scale. In terms of field recordings, we look specifically at the CA3 to CA1 input as a model system, looking both at synaptic connections, because if we're looking at communication, there are two real pathways we want to consider. One are the synaptic connections, and the other are the communications that are occurring over the axons. Now, when you consider these short connections versus the long connections, the integrated networks have long connections. And what we found is that what happens with insulin resistance, so we are able to induce insulin resistance of neurons with indinavir, which is a GLUT4 antagonist. And what you find is that indinavir, so insulin resistance of the neurons, greatly reduces long-term potentiation. But what it really, really affects is the conduction velocity along axons. So when people say they are not thinking clearly, they're not able to think quickly, there is a kind of a biophysical element to that, because if the signaling is occurring more slowly along the axons, it also makes sense that for integrated networks where the axons are much longer, you're going to feel much more of an effect. So one of the questions that I'm looking forward to addressing in the future is the question of whether neurometabolism bends before it breaks. So what seems to be the case, remember there's this kind of sigmoid here, there's a period of homeostasis which is followed by a period of metabolic stress. Past a certain point, there is a breaking point, and past that point, cell death. And so when we see the end point of that in terms of atrophy, at that point, trying to refeed neurons is not going to be successful. And so the only period in which we can actually try to achieve reversal of these effects is during the period of metabolic stress. And in fact, that's what we found. So when you look across the age span, young people show an improvement with beta-hydroxybutyrate. People in their 40s to 60s show a huge improvement in terms of their network stability with beta-hydroxybutyrate. But past the age of 60, there's almost no effect. And that suggests there may be a critical window for addressing these problems, this metabolic stress, before it's too late. So the last point I want to make is that thus far we've been talking about networks, and that's the way that neuroimaging typically thinks about what the brain is doing. But of course, processing in the brain is not done by networks, it's done by circuits. And when you see these components with lines drawn between them, this is not a pretty diagram. This is actually a system of coupled differential equations, which means that we can actually run this like an electrical circuit and ask, how does this process information over time? So we spent about 10 years deriving the intricacies of this particular control circuit, particularly this part over here with the ventral stream subcircuit, and looking at dysregulation and how it relates to trait anxiety, clinical anxiety on one end of the spectrum, all the way to sort of trait recklessness and clinical recklessness at the other end of the spectrum. And ultimately, what we want to be thinking about in terms of metabolic psychiatry is how changes in energy at the very lowest levels are going to impact the regulation or dysregulation of circuits at the top levels, because those are the components that we think are going to lead to emotional dysregulation, or the signs and symptoms that we associate with psychiatric disorders. So here we can think about regulation via feedback, sort of like an autopilot. We developed the techniques for this initially in silico and then, confirmed them with data. So here's UK Biobank data again with almost 20,000 brains. And we were able to independently replicate the same circuit that took us 10 years to figure out. This was done entirely by computer scientists using these sort of automated methods. And in fact, you see that dysregulation of these different circuits associates with different symptoms that were identified by UK Biobank. So the more dysregulated they were of these circuits, the higher their scores on these different psychiatric indices. So the question that I would have is, so how do we proceed from here? So you have a way of thinking in terms of modeling and whatever that seems to be kind of useful in thinking about how systems emerge over time. What are the trajectories? What are the emergent effects of these lower level dysregulatory processes? But if I were the only person to be doing this with my lab, that would be a very sad state of affairs. We wouldn't be taking adequate advantage of the crowdsourcing capabilities of everybody's wonderful data and thoughts and so forth. And so when Jan and I spoke about this, it was in the context of really thinking beyond what we ourselves could do in terms of modeling, and rather creating a software platform that anybody could use, which includes all of you, in thinking through how you put your data into these models and then ask what are the long-term consequences of this. So what are the dynamic consequences of the predictions and hypotheses that you are making? So our software is being developed in collaboration with our collaborators at MIT and Dartmouth. It is multi-scale, multimodal, and multi-system. By that I mean it includes not only the brain, but also the autonomic nervous system, endocrine system, immune system, and metabolic, of course. There are components that are hypothesis generators, experiment optimizers, and allow for community modeling, means you can start a model and you can finish it. You can propose a model and you can test it. And in order to do so, we had to go back and ask what are the computational primitives of brain circuits. So obviously in electrical circuits, we have capacitors, resistors, and so forth, but the brain doesn't have those. And so we could cheat and say, oh, let's put them in, but we'd be lying, which we won't be doing. So instead, we need to go back and think about biomedically, what are those computational primitives within the brain? And so we have been starting off by modeling these small microcircuits, which are literally the processing components within the brain that are ultimately going to be building blocks of larger circuits. So here is our GUI. And so what you can see here is that you have these individual blocks. So there are different components here that are modular. Each one of these is a subcircuit. So here you can model, for example, the single neuron dynamics, which are the components of an individual block. But then when you arrange them, you can also model the whole circuit dynamics of the circuit as a whole. And of course, these are nested control circuits, so you can create blocks of blocks of blocks, and you can create control circuits of control circuits of control circuits, which is necessary for most of the applications we're interested in. Finally, the last point I would like to make is that the quality of the data matter, and very frequently people tend to focus on their results without thinking about measurement error and how that can influence what you wind up with. In particular, also, I'm interested in personalized medicine, which means that we need a much higher standard of quality for our data, because I want to be able to put in a single person in a scanner, generate these control systems models of that person's brain, and then ask how that person will respond to this intervention versus that one. This might involve dietary changes. It might involve the development of a rational psychopharmacology that is based on the particular parameters of that individual. And so what we find is that if you think about signal to noise, this is at the single subject level. So here we have 4 different scanners, a sort of gold standard 3T Prisma, another 3T Prisma, 3T Skyra, and a 7T Magneton. These are all Siemens scanners. Without our noise net denoise signal, none of them actually reaches the standard of signal to noise exceeding 1, which means it cannot be used at the single subject level clinically. And so what we did is develop some hardware that would allow us to calibrate the signal. It's basically an artificial brain that has a known signal, and then we compare the known signal to the measured signal using machine learning, learn the noise characteristics of each scanner at each time, and then use that to then clean the data. So basically it scrubs the data of the scanner characteristics. And you can see a huge improvement in terms of optimizing your acquisition parameters for single subject level imaging. And so this is the device. Again, I don't really have time to get into the details, but I do have a video that shows the inner workings of the gears and how it's set up, which we had patented this last summer. So moving forward, this is a slime mold. So why do I show this? Well, actually, what's happening here is that each of these oat flakes, these are oat flakes, which slime molds apparently are extremely fond of. And this has been arranged in a particular pattern where the center is Tokyo, and these are the surrounding cities outside of Tokyo. And so it took a very large group of well-trained engineers in Japan to develop the optimal configuration of the Japanese railroad structure. It took this slime mold less than a minute. So how did that happen? Well, the slime mold, actually, each individual slime mold is not very smart, but when you put a bunch of slime molds together, they have what's known as collective intelligence. And they use a specific rule, which is that if one of them, it's called explore and exploit. Basically, they sample from the entire search space of where there could be oat flakes. And if you find an oat flake, you send a pheromonal signal that says, go here, this works. And if you didn't find an oat flake, you send a different signal, which is, don't go here, this didn't work. So why am I showing this? Because for me, this is a perfect analogy of science, where each one of us is operating as an individual slime mold, but ultimately, collectively, we have an intelligence that goes beyond each individual researcher. The question is, how do we do as a field what the slime mold colony does so effectively? In order to do so, we need to have two signals, go and no-go, basically what has worked and what has not worked. So we need to know what is replicating, what is working, also what is not working. And we need to be able to look at this at a glance systematically so that you don't need to do a whole bunch of PubMed searches and hope that you find what you should. So all of this has led to the proposal that we have something called the Baszucki Metabolic Neuroscience Initiative, which would have three arms, the clinical, the mechanistic, and the computational. And this would act as a hub for coordinating funding and research in this area. It would also have its own research that's being done in these three areas. This would include foundations, frontiers, and technology development. It would include, like the slime mold, a community-building dashboard to monitor and disseminate new results, reproducibility, and gold standard protocols. Then we would also have an annual review and planning meeting, which functions as a conference to identify new strategic directions in a rational way rather than an accidental way, as well as a journal of metabolic neuroscience. So that is the hoped-for direction for the future. Thank you very much.